So, good evening, everyone. We'll start. There may be a few people trickling in, but my name is Nick Bamp. I'm Deputy Head of Department, and a very, very warm welcome to you. Those who's the first time here, welcome to the Yusuf Hamid Department of Chemistry. Some of you who we've seen before, welcome back. It's a real pleasure having you here because, especially at the end of a day where I think it was nicer to get here than may have been earlier today, we love talking about our science. I like introducing you to the people who do the fantastic science for which the department is so very well known. A couple of formalities. First and foremost, there is no fire drill this evening. If an alarm goes off and a recorded message asks us to leave the building, please do so via the signs that are marked by the door. They're the standard little green signs, and they will lead you either to the Union Road or to one of the ramps at the bottom of the building. We just then go around the front and collect in the big car park on Lensfield Road, and there we'll be told what to do. If you need a bathroom, the bathrooms are just outside the door through which you came earlier. Please go and use them if you wish. And if you need to leave early, please identify yourself. And at the end of the evening, we'll take you out in a little group. Otherwise, if you take a wrong, you may be here for the whole weekend, okay? And I don't think we're kidding about that. Now, unfortunately, the two people who we wish to thank won't be here till the end, but these events do take a lot of effort to put on. Tonight, in the lead up to this, the sort of scheduling, the ordering, the communicating, and it wouldn't have been possible without Caroline Reed and Diane Harris, who've done so much work and have done fantastic work for the department, communicating the incredible science we do. Also, two members of Jenny's team have helped us tonight put things together, Evan Rowe and also Sheila William. I can see Evan at the back. Look, a real thanks to them. But really what this is about, it's about science. And that's the thing that we really wanted to tell you about. And it's a real pleasure to have Jenny Zhang and her team. We've got Robin Horton and also Loris Marcel, PhDs in the group, and they're here to talk about their science. And the science that they do is really important because what they try to do is look at what biology does and apply it to looking at concepts of renewable energy, and in particular biosynthesis. Now, the biographies for our speakers today are in your little brochure. Jenny began her career on my side of the world. She did her degrees at the University of Sydney and came originally to collaborate with Professor Erwin Reisner, who gave this talk a year ago. And she came funded by a number of highly prestigious and very competitive fellowships. First as a Marie Curie, and then as a BBSRC David Phillips Fellow. Now, these are hard to get, and it's a real pleasure to have had her come and work here. And she began her independent career with the BBSRC Fellowship. What is particularly great for us in the department is that earlier in the year, she secured an assistant professorship, which means she'll be here with us, hopefully, for the long term. And her group is young, bright, highly regarded, and what they will tell you is about the science that they do. Now, this early in her career, what is remarkable about the work that she does is not only that she does it here, it's that other people in other departments around the university are very keen to work with her and her team. And that is a measure of the stature at which she's kept and how highly regarded the work is. And what I've always been impressed about the papers that she's been publishing and the presentation she's given is that in the field of biology, chemistry, engineering, materials, there have been assumptions about the way that biology does things which have served us particularly well. And we've gone on with those assumptions. The first time I heard her speak about the work, I was surprised. I had to say to her afterwards, so you're telling me these assumptions that we've assumed have always worked, no one really understands the mechanisms. And surprisingly, she said no. And Jenny's trying to solve those mechanisms. All right? This is clever science, and it's a real privilege to sort of have her telling you about this work. So Jenny and her team, and they've got these wonderful little things, which we hope, Jenny, Robin, they are going to work, aren't they? Excellent, great. Okay, there will be time for questions at the end. I mean, the idea is for this to we'll wrap up in about 45 minutes. There will be time for questions for you to come and look at the equipment and to talk to the team. But we hope you enjoy the evening, and we're really grateful that you made the time to come here tonight. So, Jenny, Robin, Boris, thank you very much. And Jenny, I'll pass this over to you.
Thank you. Okay. Good evening, everyone. First of all, I just want to make sure that you can hear me at the back okay? Yes? Okay, great. So let us know if you can't at any point. Okay, so it's fabulous to have you here. So yes, I'm Jenny. Thank you, Nick, for the wonderful and kind introduction. And my team and I do a lot of science just upstairs in this department. And today we're going to tell you about one of our favorite topics, which is photosynthesis. But before I get into that, I've been introduced, so let me introduce you to the team. Because science cannot be done, and the best type of science is done with a team of people and cannot be done by oneself. So you would probably have been introduced also to Evan and Shella, who led you into the building, but you'll hear later on from Loris as well as Robin. And tonight we're going to tell you about photosynthesis. So I know that a quarter of you would have done your net skis in Cambridge. So maybe this would be a little bit of a reminder, just a 101, photosynthesis 101. And we're going to tell you why it's so fabulous. And then I'm going to tell you a little bit about photosynthesis that perhaps you don't know about. And that is quite electric. Okay? So it's going to give us lots of electricity. And then I'm going to tell you about how we're going to use this electricity from photosynthesis to, for example, make cyborgs and create these biohybrid devices to generate clean energy. So that's what we're hoping to get out of tonight. Okay, so let's start off with a definition of photosynthesis, okay? So I think the best way to define something is just try and imagine life without it. So let's just spend a few seconds just trying to imagine what would life look like without photosynthesis? What do you think? Yeah. We wouldn't be here, exactly, yeah. It'd be hard to imagine, wouldn't it? It wouldn't be so colorful, definitely no pigment, like green pigments around. So it turns out, oh, it's dimming. It turns out what happened is that, well, the planet, the Earth, is 4.5 billion years old. And for about the first billion years of Earth's history, there was no photosynthesis. And of course, there was no one to take a picture of it, so I asked AI, and yes, great, it came out, to generate a picture of what it might have looked like without photosynthesis back then. So what you probably notice, sorry if you cannot see at the back because it's a bit glary with the sunshine, but we've got these really dark blue atmospheres, and this is from the CO2 that's been emitted by all the volcanoes. You see there's no complex life forms. There have been really simple monocellular bacteria floating around, but no complex life forms at all, and obviously no oxygen. And so what happened was that about 3.5 billion years ago, one type of bacteria at least, okay, discovered a really clever thing. It discovered a way to use really abundant resources around it. In this case, it's water, so H2O. And with the help of sunlight, which we have a lot on this side of the slide as well, with the help of sunlight, it was able to split that water into its elemental form, so oxygen and lots of protons. And from this reaction, so this is a redox reaction, is what we call an oxidation reaction, if you remember. What happens is that we can then channel all those liberated electrons to another part of the cell, where then it can use those electrons to form new bonds between CO2 molecules, so that you can get very complex molecules like sugar, cellulose, and what we call biomass. And so this is now our current form definition called photosynthesis. And so this is a really important process, as you can imagine. This was the first time that oxygen was liberated, and we know how important oxygen is. And in those days, without photosynthesis, all the life forms were they were intolerant to oxygen, because oxygen can become very reactive, especially when it's been energized. And so what happened is that when photosynthesis started occurring, it actually killed off 99% of life on Earth back in those days. But luckily, that 1% that survived was able to use oxygen as sort of like a mutagen to evolve into more complex life forms. And that's actually where we eventually where it ended up eventually as us. And so photosynthesis has an incredibly important part to play in the history of life. And so, here is an image of these these ancient cyanobacteria. So this is an example of cyanobacteria living in stromatolites. I think some of you have heard of it. This is example in Shark Bay in Australia. And Robin now is going to pass around some fossils showing fossilized stromatolites, which is something like this, except the ones that are being passed around are very young. Those are 70 million years old. These are 2.5 billion years old. And so what happens is that during photosynthesis, the cyanobacteria is able to convert some of the carbonate, so from the atmosphere, into minerals, and so they can build these homes. And you can see the layers. That's basically how active they are, depending on the season. They may form thicker layers. And so it's kind of like tree rings. You can see how they grow, so they kind of expand outwards. So it's really fascinating. So we can still see that these bacteria are very successful even today, okay, because these are 2.5 billion years old, right? So, or at least their ancestors are 2.5 billion years old. And in fact, cyanobacteria are one of the most abundant life forms today. And the reason why they're so successful is because they're able to use all these really sustainable components around them to survive, okay? Okay, so while that's being passed around, you would have seen that at the back of the leaflet there are some quiz questions. And so I thought this is a really good time to help us consolidate how important photosynthesis is. So we talked about how those really old cyanobacteria is still here today, but it also has evolved into other life forms. So for example, it was incorporated into plant cells. And so that's where you get chloroplasts from. And of course, chloroplasts is producing the oxygen that we consume, and it's how we were able to breathe today. So one of the questions was, how much oxygen is consumed by an adult per year on average? And I gave a little hint on your sheet. And for those of you who did do your NatSkis, maybe this reminds you a little bit of the entrance interview questions, where it's like an estimation question. So I thought that would be a fun way to help you remember your time in Cambridge. But really, if you did do the calculation right, the answer should be B. Okay, so come argue with me if you don't agree, but later. But what that's equivalent to is 8 trees, essentially, about 8 trees producing same amount of oxygen per year. So it's a really good idea to keep trees around us and not cut them down. Okay, so today, photosynthesis is really important for many reasons, not just because they keep us alive. We can see here is a map of where photosynthesis is occurring. Okay, this is a map taken from NASA. And you can see that on the land, where the green is, that's where plants are, trees, etc., forests. But you can see there's a huge amount of bright blue and green, and that's where all the photosynthetic sea organisms are. So the cyanobacteria I was talking about, algae, dinoflagellates, which we'll hear about later. And even though it's such an important process on Earth, as Nick says, there's actually a lot unknown about photosynthesis. And this is problematic because when it goes wrong, so for example, some of you, at least when I gave this talk last year, came up and told me about how there's these harmful... Yes, it's really such a reaction. Yes, these harmful, very harmful algae growth that is in some of the Scottish lochs, for example. And then, of course, there's coral bleaching that happens. We don't understand why, something to do with how photosynthesis is going wrong. And on the other side of the coin, we need to understand photosynthesis better so that we can use them better. So as we all know, we rely on photosynthesis for food and farming, but also for our energy when we're developing clean energy. So today, we're just going to give you 2 stories about what we're doing. We're going to show you how our research impacts on understanding coral bleaching, and also about how we can perhaps develop new technologies for clean energy. Okay, so let's go back to how photosynthesis works. So I'd already told you about this process. Okay, we have oxidation of oxidation of water, and that liberates a flow of electrons. Okay, so this movement of electrons, of course, is, by definition, electricity. And so what underpins all of our research right now is based on this effect that actually photosynthesis is electric. So let me show you what I mean by that. Okay, so if you take any photosynthetic organism, including the spinach you would put in your salad, and you kind of zoom in with a microscope, you will see something like this. So this is the chloroplast, is the green parts, and inside of it is an organelle called a thylakoid. You can see it's highly folded, and if you zoom in a little bit more, you've got this amazing chain of proteins. Okay, I'm gonna really zoom in now. Now, I don't want you to be distracted by all these symbols and letters. All I want you to understand is that the magic starts with this protein here, which is called photosystem 2. It's an amazing catalyst, it's an enzyme that does the water oxidation process. And this is an animation produced by one of our students, Murray Ayres, who is a very talented filmmaker, and she made this animation for us. So you can see, basically, you have the water oxidation process here. It liberates a lot of electrons, these are little dots, and then they're passed along, they flow along this chain like electricity, and then these dots, these electrons, are used in lots of different reactions. So one of the pathways is that can go to fix CO2, so make those really complex sugar molecules. It can also go to do other reactions, forming bonds to make new molecules. It can go back into the chain sometimes, and occasionally it can escape the cell itself, so it can actually go off and do something else. And that is actually a really mysterious process that no one understands why. Why would the cell be giving away electrons? But it's also the process that we love to understand the most. So basically, what it means is that if we can put an electrode is just something, a material that is very conductive, that can receive electrons, next to living organisms, living photosynthetic organisms like cyanobacteria, then we can start to measure those escaped electrons. And I want to show you this. Okay, so hopefully this is going to work. So this is an experiment. Oh no. It might, yeah, okay. So maybe I bumped it as I was moving this. So what you are seeing here is current, and what you're seeing is the current that we're measuring over time. It should be a flat background. But of course, whenever you're doing demonstrations, something like this happens. But what we're going to do is I've got an electrode with some cells on top, some living cyanobacteria cells. It's about the size of, I don't know, a small penny. And I've just filled that electrode with cyanobacteria cells. What I'm going to do is I'm going to shine light on it, and hopefully it will still work. Oh, good. So what you are seeing right now are those escaped electrons. Okay, so the current is shooting up. And if you give it time, it will basically make a little shape. And then when I turn that light off, and I'm just going to give it a little bit of time so you can see, appreciate, so we get a lot of electrons coming out. But as soon as I turn the light off, and the light is here, and you're welcome to come and look at it later if you like, you can see that we have it coming back down. Okay, so the current is going back. So it's coming from escaped electrons, which is pretty amazing. But what is even more amazing is the fact that if I had enough time, and you were here all day, I would be able to show you exactly the shapes of the profiles that we get. And so this is something that we prepared earlier. You can see that actually what you see are these peaks and troughs, okay, these wiggles that come with the electrons. So it's not just a smooth shape. And so if I go back to my presentation, here we are. So what we've just done is we shine light on our photosynthetic cells. We have made these flow of electrons, we collected some of the escaped electrons, and basically we're able to measure these fixed signals that are coming from cyanobacteria in a very non-invasive way. So you can imagine that this is very similar to when we put ECGs on our cells, those electrodes, and you can then measure electrical signals from your heart, and that helps you to monitor your vital signs, right? That if the shape of these signals, it tells you if something is blocked, something is too slow, etc. The same thing can be done here, where we can non-invasively now monitor the vital signs of photosynthetic organisms, which is pretty exciting, and this is quite new. So at the moment, we're trying to understand what all these peaks mean. And so why could this be even more exciting? So here we have Loris to tell us more. Where would you like to stand?
I'll just stand there.
Okay. Yeah.
Hi, everyone. Can you all hear me okay? Great. Thanks, Jenny. So now I'll just talk about how we can use these photocurrents that Jenny introduced to look at a different problem, which is coral bleaching. So you may have all seen these kind of articles in the news, right? Coral bleaching is a massive problem, and what's happening is that it's becoming more and more common, and it's leading to the decline of corals all around the world. So we need to do something about it. But why should we care about corals? And look out for a quiz question in your booklet. Corals support 25% of all marine life, which is actually humongous considering that they only cover a small percentage of the seafloor. They're also really important for fishing, for tourism, and also they protect our coast from coastal erosion. If you put all of this into a money figure in terms of what they make and save as money for us, that would be 24 billion a year, which is, a lot of money. So let's take a step back and look at what corals actually are. Corals are colonies of animals. So this thing, how does this work? That's okay. Oh, there you go. This is a colony, and it's made up of lots of little individuals. And these individuals are brown because they contain brown algae called dinoflagellates. They have nothing to do with dinosaurs, but they did exist at the same time. And Robin will show you a tube of these dinoflagellates, and you can compare them to cyanobacteria and see how they're different. Now, these dinoflagellates are really important because they're symbiotic. So they make some sugars that feed the corals, and in exchange, they get a whole other array of nutrients. So during bleaching, the corals expel these dinoflagellates from their body, and they turn white because now they've lost all their brown pigments, right? And this is bad because if the corals stay bleached for a while, then they will eventually starve because they don't have any of these sugars that I mentioned. Now, why can't we do anything about bleaching? Well, bleaching is really complex. It's like a black box. So we know what goes into that box, as in we know what causes bleaching, and this is often things related to climate change, like higher temperatures or intense light. And we also know what goes out of this box. In other words, what the end result is, and that's the algae being expelled. What we don't know is what happens inside of this box, right? Because it's so complicated. And our aim here is to try and understand what happens inside of the box. So, how can we do that? Well, as Jenny mentioned, when you shine light on some photosynthetic microorganisms, they can produce electrical currents. And here I'll just show some photocurrents obtained from these dinoflagellates. And this is really interesting for two reasons, right? The first one is that these photocurrents could actually be involved in the communication between dinoflagellates and corals. So if you think of a neuron that sends electrical signals, right, to send a message, there could be a similar thing happening here, where the dinoflagellates are sending pulses to the corals, and maybe they can change these pulses, for example, when they're feeling stressed. And that could trigger bleaching, right? And the other interesting thing is that we can use these photocurrents to look inside of the cells, which is the knowledge gap that we're trying to address. So here I'm showing that when you change the temperature, the photocurrents change as well. So the higher the temperature, the lower the magnitude of these photocurrents. And without going into detail, this is really interesting because it's telling you how an external factor is influencing a reaction that's happening inside the cell, namely the source of these electrons, some of which come from photosynthesis. And I'll just take that analogy from Jenny again about the ECG, where you're measuring something external, and that's telling you something about what's happening inside of your body. We're trying to do a similar thing with dinoflagellates. So just to sum up this little section, studying photocurrents can help us both understand how corals and dinoflagellates communicate, and also look inside of the cell and into that black box of coral bleaching. So hopefully, this will help us understand a bit more about how bleaching works and, in the future, prevent mass bleaching events. Thank you very much. I'll hand back to Jenny.
Okay, thank you so much, Laura. So Laura's is a first-year PhD. Okay, so we now heard a little bit about why those bioelectricity is really important so that we can, study living organisms. But what about for energy, which is the other story we want to talk about? And so if we switch gears a little bit and think about, okay, how are we using energy at the moment? So this is UK's energy market just a few years ago, and I've checked, it's very similar still. And what you can see is that about 10 to 12% of it comes from what they call bioenergy. Okay, so you can see from electricity as well as from biofuels. So what is bioenergy and how is it related to photosynthesis? Why aren't we using more of it, especially when you look at it and it looks like it's a renewable energy? Well, let's dive into that a little bit more. Okay, so let's look at how photosynthesis is basically bioenergy. So if you go rambling in the countryside, you probably stumble over one of these massive sugar beets, at least I have in the past. And essentially, they're just massive beets that are filled with sugar. And those sugar is produced by photosynthesis, which occurs on the leaves. And I've already told you about how photosynthesis works. So this is the photosynthetic electron transport chain that we showed you before. So what happens is that during photosynthesis, we form a lot of the sugar, which is coming from the carbon fixation reaction. Then when you get the sugar, the cellulose, you can either combust it to make electricity, so heat and electricity, or you can use those sugar, isolate it from the cellulose, you can ferment it, and then isolate it some more, so lots of downstream processing is what they call it. And then what happens is you get the biofuel, so the bioethanol, for example, that you can put in your car. Now, the efficiency for that overall, for the consumption of this bioenergy, is really low. Okay, so it's less than 0.1%. So the take-home message I want you to get out of this is that overall photosynthesis, the machineries within it, the photosystems, the catalysts, extremely efficient, very high performing. But when you look at the overall process of photosynthesis, especially for bioenergy production, it's extremely inefficient. And that is why it's actually a very controversial way of producing energy. Okay, so you can see lots of pros. It's very scalable, it's carbon neutral, it's very easy to do. You just do more agriculture. However, it's very inefficient, which means you need to do a lot of it. Okay, it's competition for food farming, and if we don't have enough land, what does that mean? We basically have to take down more forests, which is what happens in Brazil, and then you get loss of biodiversity. So it's very controversial for this one reason. And because of that, there's a whole area of science that's basically trying to replace bioenergy using this topic called artificial photosynthesis, which if some of you came last year to this talk, was already given by, a very good talk given by Owen Reisner. So this is a fantastic field because what it's trying to do is mimic photosynthesis, but not using any of the biological components. Okay, using the metal oxides, etc. And you can see here are some artificial leaves. And what happens is that, again, we have the oxidation reaction. We can get out electrons, and we can put those electrons into new bonds to form, for example, hydrogen. So this is basically artificial photosynthesis. And there are a lot of really great things going for this particular field. So for example, we've got amazing light harvesters now, which means that we can absorb much more, we can utilize much more of the solar spectrum than we could using natural plants and pigments. And also you can see it's very straightforward, right? So basically we have one oxidation reaction here that goes to another reaction. No real obvious diversion of the electrons for other processes that would be used, for example, inside of a cell. Now, the problem with this field is that they don't have very good catalysts. And catalysts are very, very important for fuel formation. so for chemical synthesis. And when I talked about the energy market, how there's the way that you use electricity and the way that you use fuels, which one is the dominant one? It's actually the fuels. So we actually have the majority of our energy use is in the form of fuels, and we don't have a good way of producing fuels. Okay, so let's get into a little bit more detail, because I feel like I want you to appreciate why catalysts are so much better in biology. So we talked about how photosystem 2 in photosynthesis can do this water oxidation reaction, so it breaks up water. Water is a very stable molecule. So how is it able to do that? So I'm going to use this analogy which I used for a long time. So imagine if I give you a can of beans. Okay, it doesn't have any pull rings, so you have to open it by your bare hands. How easy would that be? Probably not very easy. You probably, dent it a few times, you hit it against the table, but you're not going to open, right? So what happens if I give you a can opener? So that is the catalyst. Suddenly, you needn't use much energy at all. It will happen very easily. You'll be able to open that can. And so basically, the photosystem 2, in this case, is like the can opener to open up the water. And then there are lots of other types of machineries inside living organisms. So here's another type. I won't overwhelm you with the name of it, but essentially this is the opposite, where it can take electrons and clamp down 2 bits of molecules, inject in the electrons as glue, so kind of like this clamp and glue gun system, and then you form a bigger, more complex molecule. And so there are lots of biological enzymes like this as well. And so in the photosystem 2, so I'm now showing you the structure here, what happens is that if we think about it like a can opener, it has all these really elegantly formed channels where the substrate, which is the water, can go in, and then that channel holds it still in the right position, and then it cleaves those bonds so that it can separate really elegantly. And this is the cleavage point. This is just basically some metal oxides that's embedded within the enzyme. So if now we go and think about the synthetic system, so if there are some chemists around, essentially we can synthesize lots of molecules to mimic the catalytic part of the enzymes. Like here, we have lots of mimics. But you can imagine them, if you compare that to a can opener, you have either a butter knife, so you can open a can, but it'll be quite messy, you'd probably take a lot of energy, or it'll be like a machete, where essentially you'll be slicing off your fingers and everything else that's nearby. So it's not very selective. So compared to a biological enzyme, or catalyst, it's not as selective, and also compared to a living cell, it's not self-regenerating, so there's a lot of things that are missing. And so this is why we have basically come to a new strategy, which is that we don't just use biology or just materials. We want to cherry pick from both and work together and see whether we can bring the best of both worlds together. And these are these biohybrid systems. And so we move on to the cyborg part of the talk. So here we can see is a lab in Berkeley. Essentially, they're introducing light harvesters to living bacteria so that it can do artificial photosynthesis, but using living systems. And here's another example here from our very own university with our collaborators who have used these cyanobacteria to drive fuel cells, essentially using those bioelectricity I was talking about, to power a microprocessor for over 2 years. So that was pretty amazing. Now, the problem is with those, the current that you use to power those microprocessors is not very much. So you can see that this is the current that we're talking about that's leaking out of the cells. And this is the level that they're at. And this is when I started working in the field. They're very low. They're low, but they're enough to power sensors. So that's fine. But if we want to go and use those electrons to make fuels, to put it to good use, we need a lot more of it, okay? So we need it to be around here. So that's about an order of magnitude higher. And that's because if we can reach, for example, the bottom threshold, then we become competitive against bioenergy, the traditional form. And if we can get even higher than that, we can compare ourselves against solar cells, which do a different function, but they're very efficient. And so one strategy, so how do we get to those? How do we increase the amount of current we get out of those living cells? We need to steal more electrons. So we're going to tell you about two strategies that we've used. One is to basically shuttle out more electrons. And what does that mean? So here we have a living cell. We have those electrodes that are collecting electrons. What we could do is introduce some shuttles. So you can think of it like an actual bus shuttle. Okay, so we're trying to send these molecules into the cells so they can pick up electrons from somewhere at a target, and then they come out of the cell, and then we want them to deliver electrons to the electrode and cycle. So like a shuttle, we just want to add more shuttles in, and that will boost our photocurrent. And you can see the shape, there's many different types of electron shuttles available. But now the real challenge is knowing where to basically design these shuttles to interact with the right target inside of the cells. And so this is where Robin comes in.
Hello, I'm Robin, and I'm a PhD student in Jenny's lab as well. And my work is kind of focused around finding these shuttle stations, because if we can find the shuttle stations, then we can make better molecules to be the shuttles, if we understand how they interact with photosynthesis. And actually, to find these shuttle stations, we don't use electrochemistry, like Loris was talking about. We actually use light to investigate the cells and the systems that we're working with. So I'm going to head back to the beginning and ask a very fundamental question of why are plants green? So the reason plants are green is because, in the case of our lovely spinach here, they're hit by sunlight, which is made up of all the different colours of the rainbow, and they absorb every single colour apart from green. And that green is reflected back into your eyes, and that's why you see them as green. Now, we were talking about pigments earlier, and the pigment that does this, absorbing and like the reflecting of the green, is chlorophyll. So these are some chlorophyll molecules up here. And just to kind of show you this pigment absorbing, Loris very, very kindly extracted some from spinach for me earlier, which we have in bottles here at the front. So if we take this chlorophyll and we shine UV light on it, so that's ultraviolet light, then hopefully you'll be able to see this at the back. Wait, Loris, can you hold one up?
Oh, sorry.
Hopefully you'll be able to see this at the back. You can see a slight red ring at the bottom there. And so that's because the chlorophyll is absorbing that ultraviolet light, and I'll explain why it's red. So when that ultraviolet light shines on the chlorophyll, what actually does the absorbing is the electrons in that chlorophyll molecule. Normally, electrons in molecules are just kind of hanging out, they're chilling, but if you shine UV light on them, they get very excited and they get full of energy. And over a very short amount of time, they lose that energy again. And when they lose that energy, they lose it in bits and pieces, and they give out light again. And in this case, they give out red light, and that's why you can see the chlorophyll glowing. But it's just a really good showcase of the fact that these pigments are absorbing light. Okay, so, no, that was my best slide transition. Okay. What about in cyanobacteria? So we took this chlorophyll out of spinach, but where inside the spinach and the cyanobacteria does it live? So if we hark back to this wonderful photosynthetic electron transport chain that Jenny was showing you earlier, and we zoom in on photosystem 2, our really great catalyst that turns water into oxygen. This is effectively what the protein looks like. It's a big blob, and it's full of lots of little smaller blobs. And the thing that matters here is that all of those green little smaller blobs, those are chlorophyll molecules. So they're all embedded in photosystem 2. And when we shine light on photosystem 2, those chlorophyll molecules, just like they do with the UV light, they get excited, and all of their electrons are running around full of energy, essentially. And in a very short amount of time later, that energy gets channeled to the very center of the protein, and that's the energy that's used to split water and turn it into oxygen. So, how can we see this happen? Because this is all to do with absorbing light, we can actually use a very fancy technique called spectroscopy with lots of fun lasers in the physics department to investigate how these photosystems absorb light over time. So we can see this transition happening using these fancy lasers in the physics department. And so then we can use this to find out how the shuttles, if we add them into the system, can change the energy transfers that we see here. And if they change the energy transfers, then we can figure out where they're interacting with photosynthesis. So, photosystem 2 with a shuttle. So we shine light on photosystem 2 again. Again, all of these chlorophyll molecules get really excited. But what we found out was that when we add in a shuttle, instead of that energy being channeled into the center like it was before, it actually transfers onto that shuttle. So our shuttle is now full of excited electrons that were previously on those chlorophyll molecules. And it can then head off back to the electrode and deliver those electrons to the electrode. So that is a pretty neat, neat set of experiments that figured that out. And what I've been working on is kind of a follow-on from that, where I've been trying to find the exact point on this big blob of protein where that happens. So I've done some computer modeling. Oh, wait, hang on. How fast do you think this happens? So this is one of the questions in your book. This happens on quite fast timescales. So we have, do you think it happens in 1,000th of a second, a millionth of a second, or a millionth of a millionth of a second? The answer is it happens in a millionth of a second. That's a picosecond. That's 10 to the minus 12 seconds. So that's how fast your shuttle is stealing electrons from photosynthesis. I'm going to rewind slightly to the fact that I've basically been working on finding the exact site where these shuttles dock onto these proteins. So this shuttle station, if you were. And we think we found it maybe. Still working on this. Please don't take this as like actual factual truth. But we have a very nice shuttle bus station, essentially, on photosystem 2, where our pink shuttle comes in right next to a chlorophyll molecule. And that's how those electrons are transferring over from the chlorophyll onto the shuttle. And knowing this, if this turns out to be true, this will let other people in the lab and other people down the line design better shuttles that can fit into that pocket even better and more efficiently steal the electrons and take them to the electrode.
Okay, thank you, Robin. All right. So thank you so much, Robin. Robin is a second-year PhD student here. So, okay, so now, oh, yes, and this is on the front of your booklet. Actually, Robin drew this figure. You can see that these are shuttles stealing electrons from the photosystems. Isn't that amazing? Okay, so I'm going to, in my last 2 minutes or so, because I know I'm running out of time, is to tell you quickly about our second strategy to boost those current. And it's a very obvious one, perhaps, is how we design these electrodes. Okay, these are the electrodes that receive the electrons from the cells. So imagine if we can redesign them so that they can accommodate as many cells as possible. That means we have more shuttle stations to dock and steal electrons from. It's pretty obvious, but actually, it's a question that we thought about. Well, how are we going to answer that? Like, what is the best structure that we could design? It's almost like an architectural question, if you think about it. And so traditionally, people use very simple, flat electrodes to interface with the cells. But you can also imagine more complex structures. So these are like porous, conductive electrodes that are like sponges that can sponge up a lot of cells. And then you can think of other architectures where it's more biomimetic, like trees. So which one of these, for example, we've explored, do you think would give rise to the best photocurrent? Well, the first thing we need to do is to make these electrodes. And so part of my lab actually does a lot of engineering. So these are engineers here, Lingying Shan and Xiaolong Chen. And what they've done is they developed a new technique in which, and you can focus on the crosshead here, where we can 3D print lots of different types of electrodes. This is just one example, with lots of fancy structures. So here are pillars with branches like trees, very quickly. So you can see that this is basically making one electrode in 2 minutes or so. So that's pretty incredible. And what that means, is that we can get now a big library of electrodes. We then can incubate these electrodes with lots of cells and see how many cells can fit in. So these green parts are where cells are fitting in. And then we can test them. And this test station is very similar to the one we have set up here, where we basically shine them with light and then measure the photocurrent. And so, just to cut a very long story, very short actually, I'm going to give you the results. I'm going to summarize to you our results, okay? So we have these 3 electrodes, 3 types of electrodes. We're going to imagine that they're real estate for our cyanobacteria. Okay, so I can tell you that if we think about the square footage wise, the square ones have the lowest square footage, and then the porous electrodes, you can imagine it's kind of like, so if the flat one is like a bungalow, okay, the middle one is more like a big mansion with lots of rooms, and then the bottom one is a little bit like a skyscraper where you can get very high into the sky. And so then if we think about, okay, so basically our macroporous, I mentioned, has the highest square footage. And then we can load how much cells can we squeeze into these architectural electrodes. Then you can see that with flat electrodes, we can't get very much in. We can get a lot into our porous electrodes and fairly good into our skyscrapers. And now we're going to think about light, because obviously cyanobacteria needs light. So number of windows. So of course, with a bungalow, you just have a wide open window, so it's very high. Microporous ones, has not very much high windows. Some of them are inside, so there's no light. And then with our pillars, again, very high, so pretty good. So this is probably going to be the last quiz question I'm going to give you. Which one of these do you think would be the best for getting photocurrent, so is going to give us the most productive cyanobacteria? Who thinks the flat one? Anyone? Who thinks the porous ones with the highest loading? Yeah? Yeah, very high loading, yeah. Who thinks the tallest ones? Okay, well done. So I didn't make it too hard. So yes, you're right. So even though we can squeeze a lot of cells into the sponge, because of the fact that these pillars can balance everything, so you can see it can relatively high cell loading, relatively high light transmission, it actually gave us the best results. And so this is a type of methodology we're using to try and work out what gives rise, how can we steal more electrons, how can we get more current. And so if we now go back to our plot, okay, so this is where the field was a few years ago. When we apply those shuttles and when we use these new 3D electrodes, where are we now? Can we get close to this target area? Well, the answer is yes, we can. We just now stepped over that barrier with now 10-fold enhancement, and that means we're actually outperforming the traditional bioenergy methods in terms of efficiency. So this is a really great step forward, is a really great proof of concept. And we've done calculations. We know that you can get higher. So this is what we're working on. And you can see that this was only progress from 2 years ago. So we're really working hard on trying to push this as hard as we can so that we can make this even more efficient. And so I talked a little bit about, energy conversion, and Laura's told you a little bit about monitoring climate using our platforms. But we're doing electrochemistry here, and we're combining with lots of different scientists. And the platforms that we're developing can actually feed into many different other areas that we can't even imagine right now. But here are some examples. So the cyanobacteria we're talking about is really good at CO2 capture as well. So you can imagine we can use electrochemistry to boost that. If we can squeeze more electrons in, it can fix more electrons. So this is something we're also interested in. And lots of other lessons as well, even feeding into healthcare, which I won't have time to talk about, but basically it's really exciting directions that we're taking. So we're now at the end. I just want to give you some take-home messages. Okay, so we talked about why photosynthesis is really important and the reason why we're here today. It provides us with food, oxygen, etc. But even though it's so important, there's still so much to uncover in this field. There's just so much unknown, which is incredible. But what we do know is that there is amazing chemistry in there that we can learn from, that we can use. And these we can imagine them being fed into clean growth technologies. Example here. Okay, so with that, I'd just like to thank everyone in my team again. So if you want to ask any questions, we will take some. But so Evan's floating around. He also works with cyanobacteria and the bioelectricity. And Shella also designed the shuttle. So if you're interested, they're around. Oh, and of course, Loris and Robin are also around. So thank you very much for coming, and I hope you enjoy our little lecture.
Well, I hope you'll agree that that's wonderful science, described eloquently and thoughtfully. And it's something that Jenny has been great at, and I think she's really passed on to her junior colleagues, is the ability to describe science clearly. Because at the end of the day, if you don't get it, what's the point of us doing this? And I think the slide that's very important is a slide, go back to your one with it that's got the little star. if you take anything home, I think this is the one to remember, because over the number of years, look at that improvement, right? And that's done here. So if they are to push this, and when they appear in the next BBC thing, or take that special flight to Stockholm in a couple of years' time for a Nobel Prize, you remember that you saw the evolution of this graph. So really, really wonderful work. So there is time. I know a lot of you do realize that what you're drinking is not alcoholic, right? Excellent, right? Because it'll be a safe trip home later. We do have time. Look. I'm sure the team will be happy to answer questions. Do we have, oh, hi, Diane. There is another microphone, or should I just try and pass this one over?
I think you just pass it over.
Okay, or we'll try and keep an eye on time. We'll try and get as many questions done as possible. If you're too far back, I may ask you to tell me what it is, and I'll repeat it here. But
so there are clearly billions, if not trillions, of dollars of profit to be made by exploiting this. If you can do green hydrogen much, much cheaper than other things to power steel and do all sorts of things. What is your strategy for working with partners to commercialise this so that we in the UK make some of that money, so we can do more research instead of gifting it to the rest of the world?
Yes, I think your question is a really good one. It's about implementation and how do we bring this science that's done in the lab to actually usable technologies. So a really important part of this is actually us identifying partners in industry who are already doing something similar. So we have, for example, a partner that makes food from food ingredients, from coupling it, microorganisms to electrodes. So by partnering with them and understanding what they need and what kind of metrics they need, then we can solve questions from the basic angle, but also address what they need in terms of like what kind of bottlenecks, etc. And that would help basically launch this technology into the market quicker. It's still going to take a long time because there's so many hurdles, regulations, etc. But by working with industry, essentially, is the answer.
I may sort of pass some of the answers to your team as well. So the next question, you had your hand on next.
Hi, so question on for Robin. So do you think the shuttles in many ways are a combination of the sigma star transition, and also you use this spectroscopy-based measure the different transitions within the structure?
So
Without doing undergraduate chemistry, I'll get Robin to try and do that in a couple of minutes.
I would like to preface this with I didn't do undergraduate chemistry. I'm a physicist and also a biologist. Was it about like a sigma star transition?
The electron transition, yes.
Yeah, so we actually don't know what transitions are happening. It isn't energy transfer. Because the excitation and emission spectra don't overlap between the shuttles that we have and the chlorophyll molecules. So it is straight electron transfer. But I did try and model it once and I was very unsuccessful. So I don't actually know. It would require a lot of density functional theory calculations that I don't want to do myself. Yeah, they have very complicated electronic structures, and if you want to come and do the density functional theory for me, please do.
Could I say something with that? So actually, yes, it's really complicated, and we've started a collaboration with David Wells from the department who exactly addresses this question. So hopefully in a few years we'll be able to tell you.
Now, yeah, if you could, Evan, if you could take it a little bit further back, just because I think we should throw it.
I really enjoyed your presentation. How long do your electrodes last before they begin to degrade?
Good question.
Yes.
Evan, you can answer on the way.
Yeah, maybe you can answer it. it's a great question. So there's so many different components to these electrodes, so the biology, and then there's the abiotic part, so the materials part. So the biology we know can last a long time because of the fact that they are living cells. That's one of the beauties of them. They can self-repair, they can self-recycle, but obviously the abiotic part, the materials part, cannot do that. So really that is the limitation at the moment. And so with these type of structures or these type of materials, indium tin oxide, it really is down to the structural integrity then. Then we have to rethink how we design it so it's more strong, so that it doesn't fall over or crack or things like that. But in terms of how long they last, it depends, basically, is what I'm saying. But all I can say is that for example, the electrodes that are used to power those microprocessors, they've been going for 2 years now, and it hasn't stopped, and those cells are very much happy and still giving away electricity. So I think, you know, we're still measuring it, or our collaborators are still measuring it. So many years, we hope, yeah.
I have a question about the coral bleaching. Is it working?
Yeah, it is.
I'm afraid I'm a computer scientist, so I don't know much about chemistry or biology. Just give me the power for my chips and I'll be happy. If I was going to look at a black box system, I assume, if it was like a software system that I can't see the details inside of, I assume that somebody designed it that way for a purpose. Now, I'm not suggesting that somebody necessarily designed coral bleaching, but what if we made the supposition that the process which resulted in coral bleaching existing did it for a reason? As in, then there may be a situation where what goes into the coral bleaching, like the rise in temperature or whatever, once upon a time was a good reason to expel the bacteria. Would that type of investigation help at all or not?
Yeah, should we give it to Loris? Loris, would you like?
Yes, that's a really interesting question. I need to think about it, actually. But I would say bleaching is actually not as bad as we always say. So it's a natural process, and actually corals can recover from it. The problem is when it's prolonged, especially nowadays with more and more climate change things. Yes, it might not necessarily be a good idea to prevent it, but by understanding the mechanisms that make it up, we can try to limit other things inside the cell that can then, stop damage from happening. So it's all about really understanding what's going on in the first place. But yeah, I see what you mean. Yeah.
Okay, so I think we're the broadcasting is about to end anyway, and it's a good time also to stop so you can come and chat to the team, look at the equipment. So look, a thank you to everyone concerned, to Jenny, to Robin, to Loris, to Evan, to Caroline and Diane, also to Nathan and Michael who are recording this so that other people can see it and also see it another time. So thank you to all those involved. Thank you very much for coming. Please do come up and talk to the team. There's a little bit of time, and as we said earlier, we will lead you out so you don't get lost. But have a safe trip home, and we hope to see you next year for even more science. So thank you for coming, and thanks to all the people involved.